Hi, kiddos. Welcome to your first virtual class. So as we had discussed before, we've completed the content for AP Biology, but there's a few details, some little items that oftentimes show up on the exams but aren't considered to be testable material. So I wanted to go through those items with you. And as we go through this PowerPoint presentation, I'm going to try to keep it quick. I know there's a lot of slides. I'm going to keep it quick. But as we go through this PowerPoint presentation, I'm going to be challenging you to answer a few questions as we go, which may become discussion topics for later Zoom classes. So you probably remember that we talked about the origin of the cell and that life started about 3.6 billion years ago. And it started with just a phospholipid bilayer and some maybe protein folding prions inside doing basic metabolic processes. They also think that RNA may have been the first molecule of inheritance instead of DNA. So I challenge each one of you, this is your first challenge, I challenge each one of you to give me one good reason why RNA may have been the first molecule of inheritance. As cells became more complicated, then, of course, their metabolic processes became more complicated. The next really big advancement for cells was endosymbiosis. So think a moment. Do you remember what endosymbiosis was? If you thought that's when one prokaryote ingested other prokaryotes and then they lived in symbiosis in a mutualistic relationship together, then you are absolutely correct. Can you, second challenge, can you give me 3 reasons that we think that mitochondria and chloroplasts were once individual free living cells? One of the great things that's going to become apparent as we look at the development of animal and plant systems is that there were huge events on our planet that helped to shape the selective pressures that selected for specific tissue types or for specific advancements. And this right here is the penetration of our atmosphere by a meteor. It was a massive meteor that was said to hit the Yucatan Peninsula. And it's been somewhat associated with the extinction of the dinosaurs. And so that would have created a massive selective pressure. So they say that the smoke and the wood, the dirt and the dust was enough to shade out the planet, and it changed the plants that were living on our planet. It also made the planet a little bit colder. And because the planet was colder, mammals were given a selective advantage, and the birth of animal diversity came from this massive, we would call it a catastrophic collision. With every cyclic warming or cooling event that our planet has experienced or significant change in atmosphere, we've seen a increase in extinction. In fact, we think we may be in the 6th mass extinction. And what that does, as you know, when the environment changes, you have a new set of selective pressures. You have new niches open up that may not may have been filled by other organisms before. So we're in this really interesting and dynamic changing point of our planet's life. So that may lead to the question, why are some organisms more adapted or better adapted to live in those new sets of conditions than others? So we're going to pull together a lot of ideas as we discuss this and we talk about the evolution of systems. But I want you to remember that all of our expressed traits are coded for in our genetic material. And how long proteins are expressed can alter either how long you grow. So for instance, how long human growth hormone is being generated can impact how tall you are, and how long certain proteins are activated during fetal development can impact your bone growth. For instance, you can see here's a chimpanzee fetus and a human fetus, and you can see that the bone structures are very, very similar, but during the baby's growth, that different genes are activated at different times. They may even be the same genes, but if they're activated for a longer period of time, you're going to have overexpression in one organism and underexpression in another organism. And that can lead to what we call the phenotypic or morphological differences. Remember, phenotype was face, it's what you see. And morphological, like if you think of like a morphose, or I'm going to morph into a dinosaur, I don't know. But if you think about morph, it means to change body shape. So when we're talking about morphology, we're talking about what's actually seen on the outside of the body. The term used to describe this difference in time for gene expression, which leads to different phenotypic results, is heterochrony. So hetero means other, chrony means time. And so that's the term that we use. I would probably write that down, just saying, maybe a good idea. So one of the things that can impact an organism's morphology, or how it looks, is when it doesn't lose developmental characteristics as a mature adult. And that's called paedomorphosis. Take home term, stick it in your pocket, write it down. Paedomorphosis, paedomorphosis. And pretty much that's when the adult organism maintains, for whatever reason, a immature characteristic. So you can see that this salamander, which usually salamanders lose their gills during their developmental process, this one has maintained it as an adult, making this salamander unique and different. So there are a set of regulatory genes which are known because they regulate the expression of other genes and can dictate how long the gene is turned on, whether or not the organism shows the mature characteristic, and even determine things like, where is that gene going to be expressed? Is that arm going to come out of the head, or is the arm going to come out of the shoulder? And these body planning genes are also called Hox genes, H-O-X, Hox genes. That's a take-home term, put it in your pocket. So you may ask, hey, Mrs. P, what's up with the stickleback fish? For whatever reason, in the last few AP exams, the stickleback fish have shown up, and this experiment in particular is often referenced. So I wanted to make sure that you were familiar with the experiment. So pretty much the marine populations of the three-spined stickleback fish have a set of protective spines on their lower or ventral surface. But in lake populations, so here's the spines and the ventral surface, in the lake populations, you don't see those spines. Now, because the two populations are thriving in two different environments, it's very possible, and we did a whole experiment on this, but it's very possible that those spines give them a selective advantage. So some scientists were looking at these differences in fetal development and then the later adult morphology, or how the adults looked. And they isolated it down to the expression of one type of gene, the PIDX1 gene. And they found that, as you can see here, that there was no difference in the gene. There was no mutation that caused this. But what it was a difference in the regulation of that gene, right? The Hox gene's determining how long it's going to be expressed for and where it's going to be expressed. So in this case, you can see in the fetal development, you have the PIDX1 gene being expressed in the mouth, but also down here in the pelvic spine area. In the lake stickleback, you can only see it being expressed in the mouth. So because this PIDX1 gene is changing body plan, we would call that a Hox gene in a general term. It is a type of Hox gene. In today's lecture, I gave you 2 question challenges. And the first was, what evidence do we have to support the idea or the hypothesis that RNA was the first molecule of inheritance? So I'd like you to look that up. The second question challenge was, what evidence do we have that suggests that mitochondria and chloroplasts were once free living cells? And then I gave you 3 take-home terms that I want you to understand and know and put in your tool chest. And that was heterochrony, paedomorphosis, and Hox genes. So I need you to know what all three of those things are and have an example of each for